Okay, so now we're going to have our second talk on Alzheimer's. And so Pierre Mourad from the University of Washington is going to give that presentation, and pretty excited to hear this. So take it away, Pierre.
Thank you. So Pierre Mourad, University of Washington. I have many affiliations. I'm blessed, and I think of myself as a neuroengineer. Even though I'm in neurological surgery, I have a PhD, not an MD, but I collaborate with my MD colleagues. I wrote a paper, review paper, now 7 years ago, before anyone had studied nitric oxide synthase as related to ultrasound in the brain, saying, gee, I think this would be a good thing to do in the context of cerebral vasospasm, also good for stroke. And Gucci and his friends showed that it's relevant in the case of their approach to delivering ultrasound to brain and increasing cerebral blood flow, hence some dementia histopathology and behavior. So a very interesting paper came out of MIT a small handful of years ago. They noted that in the case of dementia, there's a reduced brain activity in the gamma band, let's say the 20 to 50 hertz band of brain activity. And they wondered if they increased it, whether they could address diseases such as Alzheimer's disease. In a variety of different ways, they exposed mice that produced human amyloid beta plaque in them, so-called 5XAD mice. And my favorite version of the experiment is they put these mice in a box and they flash light 40 times a second, which is in the middle of the gamma band. And they looked at the brains, in particular the visual cortex of these mice that were exposed to essentially a disco flashing at 40 times a second. And they found in only the visual cortex that after a week, the amyloid beta plaque burden reduced by about 50% through a mechanism whereby the microglia became activated and physically attacked the amyloid beta. Since these mice were awake, the main part of their brain that was activated from the light was the visual cortex, and they did not see this anywhere else in the brain. And since I know there are human studies right now trying to play with this kind of thing for people to see if they could affect the amyloid beta burden with light, with sound at 40 hertz, with tactile sensation at 40 hertz, and what have you. So I read that paper and I said, oh, I bet through ultrasound, I can activate brain 40 times a second and beyond the visual cortex, wherever I put the ultrasound. And I published a paper that shows that, and that's what I'm going to discuss right now. So we beamed ultrasound into the brains of mice, the same mice that expressed amyloid beta, human amyloid beta, so motivated by them. We put them under metatomidine, so we sedated them. It was not anesthesia because in their study, the mice were awake and I couldn't deliver this ultrasound to mice that were awake. Gave it 1 hour of ultrasound, then 1 hour of rest. I used 2 megahertz ultrasound because I like to think clinically, and it's just that much easier to get ultrasound that's near FDA approved parameters for diagnostic ultrasound through clinical IRBs. So that's why I chose 2 megahertz. So we stained for things like microglia, which is the red, for amyloid beta, and what have you, after 1 hour of ultrasound. And to give you an example, along the bottom, we have examples of inactive microglia. Think of them as having the little cells with these big arms that are reaching around trying to find stuff that's bad. And then when they're activated, they tend to ball up and like you think of them getting stronger in a sense, then reaching to specific places where there's damage. And so we analyze the aspect ratio and roundness of microglia in these slides to see if they were activated. So what we found after an hour of application of ultrasound and an hour of rest was as follows. So this is a coronal slice of mouse brain. Those are the ventricles. There's the hippocampus. There is the cortex. So the top 2 we applied ultrasound to one hemisphere, and the bottom one, we sham applied ultrasound. And what we've mapped here is the activated microglia based upon their shape and whether or not they're co-localized with amyloid beta. And where we applied ultrasound, we saw an increase in this combination of activated microglia in physical proximity to amyloid beta plaque. And this was acutely. So we said, oh, that's fun. Let's do the next part of what the MIT group did. Let's do this every day for a week and see what we see. And what we saw was, in this case, we applied ultrasound to both hemispheres. Again, we have a coronal slice. There's the hippocampus, et cetera, and green is amyloid beta. For the treated ones, we saw much less green than in the sham treated ones. Indeed, we saw a reduction by about 50% in the plaque in these mouse brains as compared to the sham treated animals. So following, as motivated by the work out of MIT, we found that wherever we applied ultrasound, within an hour or so, there was an activation of microglia, and within several days, we saw a reduction in amyloid beta plaque burden, thanks to that ultrasound.
Did you treat just one side of those, or did you?
No, we treated both sides in this case. And it was 50% across the brain. It was, if I can remember the paper right, it was something like 30% in the hippocampus. In the original study, we activated, I'll go back, we applied ultrasound to one side. For the chronic studies, we applied it to both sides. I'm looking to see now if, with different mice, if we're actually affecting tau burden, and we're looking at this in the context of sham and actual traumatic brain injury. And that's where I've taken my research at this point. I do have a device that Verisonics based, so simple. I'd like to apply it to humans. We're applying it to mice now to make sure it works, and I'm real curious to see if I could apply it to humans and see if we get essentially a plane of at least amyloid beta reduction thanks to this. And that is my very quick talk on this subject.
Super interesting, Pierre. That's awesome. And I'm glad to see that you were sharing with the students some unpublished sort of cutting edge work. In terms of the 40 hertz, so everyone in neuroscience has heard that paper, and they sort of immediately say, what's with this magic number? And you've thought about it more than anyone, I mean, of us at least. What's the deal with that?
Well, what I didn't mention from that paper, but should have, is they did the same experiment with 30 hertz, 50 hertz, 80 hertz, and it only worked at 40 hertz.
Can you speculate, or is that too, I mean, is it just really kind of a miracle number there?
I haven't read their most recent papers. I hope they explain it. It beats me. I think last I heard people going, why? I don't know. I would like to explore it with ultrasound at different frequencies, but 40 hertz works, right? So I'm trying to optimize it in other ways.
Yeah, it actually, so if the students have already seen Scott's lecture, it sort of begs the question of whether or not ultrasound is just generally good for reducing these pathologies, or whether 40 hertz was actually really good for you as well.
And now, of course, that's aim 15 in one of my proposals. So
we knocked out cholesterol in astrocytes, and it brought down both amyloid and tau. And interestingly, we haven't done a ton on this, but the brains that Kim sent, when you apply ultrasound, it seemed to decrease the cholesterol. So I don't know how it would do that, but the ultrasound seems to be counteracting cholesterol too. That could be a way.
With those acute studies. So
yeah.
like within an hour or so, the animal was euthanized and
within, well, it was fixed.
It was perfused while experiencing ultrasound.
2 minutes.
What percentage of cholesterol was reduced? Like reduced by 10%, 1%, 50%?
I don't remember exactly. I think it was more like 10% or something. And it seemed to be more in the plasma membrane than the overall total. We could probably look at that and send you that data. Cam, would that be all right?
I don't have any of the skill set and knowledge base you have. I can't steal anything that you guys are doing. So yeah, feel free. But so
we're not worried about it. We'd be excited for other people to work on this stuff with.
Well, I'm open to collaborations. Kim knows that. I'm almost pathological in my willingness to collaborate. I'm very social. So it was removed from the plasma membranes within minutes. That's what you just argued.
We were shocked. How could that possibly be? In fact, we thought the time period would be so short that how could that be? And
Well, okay,
it must
let's
be metabolized.
well, how about this? I mean, in a minute, how many cycles of ultrasound did these membranes experience, right? Potentially millions. So on that time scale, it's really long.
That's true. I didn't think it was going to change that quickly. I was surprised.
I would have been shocked. I am shocked. But I mean, the thing about the ultrasound is how quickly you're applying stresses. Within each acoustic cycle, there's a little bit of a stress, right? And you were pulsing at what, 20 times a second, I think? 10, 20 times a second. So, you know, in a minute, that's hundreds of shear stresses or stresses of some sort. So relative to the time scale for, there you go. So what's the time scale for rafts to move around, at least in a little bit?
They
Is that
move.
microseconds? They're fluid. They're fluid
Yeah.
on what time scales?
So we did that. I was going to show a video of that. They look like they're moving around on the millisecond. Well, you can only look at the millisecond time frame. They may move faster, but they move
Sure.
a lot in a millisecond time frame, and they form and come apart. Certainly in the 20 to 30, certainly in the 500 millisecond frame. Like we see them completely come apart and then reassemble within 500 milliseconds. So
Millisecond, so half a second.
yeah, but we've seen them going through multiple stages in a second. So I don't know, and I don't know if they're coming apart really fast and we just trap it in multiple times in that time frame. As fast as we can see it, they come apart and go together. They're moving around all the time. They're dynamic.
It might be faster. And again, the time scale of ultrasound, as you know, is really in its fastest is microseconds. And depending on how long your pulse is and how many pulses, it's milliseconds at a minimum. So for the life of some poor little APP protein sitting on a raft, they've been buffeted hundreds to thousands or more times. And maybe that's the way to think about it. Or it is a way to think about it, excuse me.
Yeah, somebody needs to figure out that that magic, like really what the molecular basis of that 40 milliseconds is.
Yeah, the 40 hertz. Yeah, it's
yeah, the 40 hertz. I mean,
for those in the audience, I think we've given you examples of why science is so fun and why social part of social science is really important.
Yeah, the value of collaboration and talking to each other.
Yeah,
okay, so I think I'm going to turn our recording off at this point on that positive note.
Yeah.